Welcome everybody to our today's episode of Phenomics webinars, and welcome to season 2, kicking off today with two speakers that we have here on the topic combining 3D and gravimetric data from plants to breed climate smart crops. Phenomics webinars is being brought to you by the European Phenotyping Research Infrastructure Emphasis, by the International Plant Phenotyping Network IPPN, and the European Infrastructure Access Project EPPN 2020. Please welcome with us here today CEO of the company Phenospex, Grégoire Hummel. Welcome, Grégoire. Yes, it's really that guy in the window here. If you compare him to the photo on screen, you might as well think it's two different people, but no, it's really him. It's just 10 kilogram more. Grégoire, welcome.
Yeah, the picture below, it's 10 years ahead. So it's looking into the future, actually.
Yeah, I'm crossing my fingers on that. Let me quickly introduce you, Grégoire, to our audience. Grégoire is a plant scientist by origin. He was born in the 70s in the beautiful city of Regensburg in Germany, closely followed by his diploma in 2003 at the University of Ulm in plant physiology. His thesis back then was on, if I remember correctly, and if my notes remember correctly, on growth, response, and allocation in an epiphyte and its host plant to different light intensities. Is that correct, Grégoire?
Yeah, that's correct. A lot of manual phenotyping.
And then afterwards, in 2008, he did his PhD, finalized his PhD and postdoc at the Institute of Bio and Geosciences of the Research Center in Jülich, Germany, on the growth dynamics of tobacco after simulated herbivory. Also correct, I hope.
Yeah, also correct. Well researched, Philip.
Thank you. Then in 2010, and this goes up to now, he co-founded the Dutch-based company Phenospex, and since then has led this company for the past 11 years already. Oh, what a time, what a long time span, as a CEO. Phenospex itself designs sensors and image analytics in order to extract quantitative metrics about plant phenotypes, health, physiological processes, and also yield of crop and other plant species. Grégoire, can you fill us in a little bit more on the details of Phenospex and what you are doing there?
Yeah, good. So do you want me also to kick off my presentation?
Go along, please. Yeah. The stage is yours.
Yeah, thank you very much, Philip, and thanks for the intro. And again, well researched. Yeah, indeed, I did some research in the tropics. I spent 1 year in Costa Rica, and there I was working as an ecophysiologist, and I was comparing different growth strategies between liana trees or lianas and supporting trees. And for that, I had I was focusing on growth, and I had to travel each weekend to the forest, which was about 4 hours drive with the bus. To measure the growth rates of my plants and my lianas, and that was quite exhausting. And I thought maybe, we should automate this process. And years later, I came then up with my partner at that time, Vladimir, and we founded Phenospex and developed solutions to help biologists like myself. And that's also pretty much what the company is doing still today. So what we are about at Phenospex is to actually push forward the transition from a very traditional way of measuring crops. What I mean with there is, like, I went to the rainforest and measuring with rulers. Today, already still in breeding, a lot of visual assessment are still done. It's enormous amount of labor and time and money then typically you have to prepare your data, to put your data in an analyzable format, which also takes quite some time. And at the end, you only have a small time fraction to actually analyze your data and to interpret and draw the right conclusions out of it and to create knowledge. And what we are about at Phenospex is actually to automate this process and reduce the time for measuring dramatically, to reduce the time of data preparing down to 0, because everything is already digitalized in the first step. And our value is basically to free up a lot of time on the researchers' end to really do their job, analyze the data, interpret the data, and generate knowledge and bring ahead the field of agriculture or plant breeding. So that's really the value and what we are about at Phenospex. Our core products, threefold. So one is a multispectral 3D laser scanner. Basically, it's a sensor which is assessing the shoot of plants. Then we have gravimetric platforms. By the way, this is a picture which is from the platform ICRISAT. And we invited Vincent Vadez, who's going to be talking about this, especially how you analyze this data. So that's these gravimetric platforms. So these are a cell. On these cells, you can position a plant, and it's tracking basically the weight of the pot. And we do that every minute, so in a very, very high interval. And since the plant is transpiring and we measure, we see how the weight is going down. And this is actually reflecting how the plant is transpiring. So get more or less real-time information about plant transpiration and understand how the plant is regulating its transpiration rates towards environments, towards chemical. So this is part of our product. We call it Drought Spotter. And then we also develop software, what we call hot control. So basically these are databases where the data is stored, managed, and we have tools to analyze the data, to aggregate the data, and to help the scientists to draw the right conclusion out of it. This is a typical setup. So this is a phenotyping or a high-throughput phenotyping machine. We call that the field scan. Vincent Vadez is using a similar platform in ICRISAT, what we actually co-developed years ago. I think 2013 we started. And this shows actually quite well what kind of data we combine. So on one hand, you have here the scanners, the sensors which are capturing the shoot of the plants. Here down there, you have the scales on which you can position the plant. And in the background, you have also climate station. And this information is gathered at the same time and can be combined and analyzed together by our platform. We just launched a new version of the PlantEye. And that's something we are really proud of. And to be honest, this is really a sensor I've always been dreaming of. And I don't want to go into too much details. That's why I copied here the barcode. So you can just make a picture and then you get to a landing page of Phenospex, and there you have really cool animations, graphics, which are explaining you all in detail the benefits of this system. And this is really, this sensor is really accumulating 10 years of experience at Phenospex, and really manifesting our leadership position as a phenotyping supplier worldwide. And have a look at that. It's really a great sensor. So what it does, basically, it's scanning your crops. So you generate a 3D model of this plant. So it's quite quick and quite high resolution. You see, we get a real 3D representation of the plant, and we also add multispectral information. We process this data in real time, and what we extract, or what you get as a user, is information about your morphological plant parameters, which are describing the height, the biomass, the 3D leaf area, which will play an important role later on in the presentation of Vincent. And also the multispectral data can be used to quantify parameters which are related to healthiness of your plant, which correlate with chlorophyll, with pigments, or with diseases. So that's really, from these 3D point clouds, you get directly to this metrics. So this is around the PlantEye F600. And then a few words about the gravimetric platforms. So here, one more time, you see these cells, in this case with some banana plants sitting on these cells. So here we track the transpiration of the entire plant. So that's a quite neat system. And additionally to the transpiration rates, you can also add actually a component here. You see this little hose. So actually these systems are also irrigating your plants. So you have a software and you can actually create a an irrigation scheme, and then the system automatically controls each pot individually and maintains the moisture level in your pot. So you can basically design an experiment, and the machine will take care of it. And I can, you know how difficult it is if you work in a drought research, and if you want to create dry downs, it's quite difficult actually to control that. So this machine is taking over this task. The cool thing about it, and the unique thing about it, is that these cells can be deployed in greenhouses, but they also can be deployed in the fields, and that's really unique. So it's really high quality components, stainless steel, it's no plastic, so it's quite robust, and it's really suitable for indoor and outdoor applications. A third unique point is that it can actually accommodate very small pots, but also pots up to 1 ton. So like this picture here, these guys are mimicking a small field plot or field situation. So actually you can also hold quite big pots as well. And that's also quite unique to our system. Yeah, so that's basically the last slide. A last advantage of our company is actually that we provide both information on the shoot with the PlantEye and information of the transpiration rate, which is quite important because transpiration should always be connected or normalized to the leaf area of a plant. So also this is something which is unique with Phenospex, and that's why we are super happy actually to invite or have Vincent here. Vincent is, we worked a lot since quite some years, developed quite some parts together with Vincent, and he recently published this publication, and I think it's of great value for the phenotyping community, especially those which work on water saving traits or on drought tolerance traits. So basically he actually continued the development and developed some new ways of analyzing the data which are coming from these gravimetric cells and show you how you can use that in breeding. So also here I added the QR code directly to this plant methods, so you can directly download this paper. But Vincent will talk now about this paper. So Vincent, hello, and really cool that you are here, and explaining your findings and how this will help the phenotyping community to improve the analysis of this kind of data sets.
Well, thank you, Grégoire, for that quick introduction. So indeed, I mean, a long history between myself and Phenospex. I mean, it comes back to the time I was, a principal scientist at ICRISAT, and a bit like Grégoire in Costa Rica, I mean, looking for quicker ways to measure leaf area. That was my first entry point into the high-throughput phenotyping, technologies that Phenospex was offering. And then after that, about measuring transpiration. So it has been a long and fruitful and rewarding journey, and a lot of interaction with Phenospex in all those years. So today I'm gonna talk to you about one trait that we had investigated a lot in our portfolio of research. Let me sort of put my presentation in presentation mode if I can. It should be now. So that trait is a transpiration restriction and the high VPD. Before I continue, I should say that I used to be a principal scientist at ICRISAT, so one of the CGIAR center. In the past 4 years, I've rejoined the French system. I'm now a director de recherche at IRD's, the Institute for Research and Development. So I keep collaborating with ICRISAT, so I keep interacting a lot with that platform that I've developed that I'm going to show you now. And I happen now to be posted in Senegal for the past months, and posted here, in fact, to think of developing and pushing a little bit the phenotyping agenda here in the Senegal system. Anyway, back to the presentation. So transpiration restriction under high evaporative demand or vapor pressure deficit, if you prefer, which is a key trait for drought adaptation and climate change adaptation, which we have been investigating in the past decade plus. Why is it important to look at plant-water relation in crop is you need to get back to the basis of plant production. It's photosynthesis. And in fact, the reason why we want to measure transpiration is simply because transpiration is a very close and good proxy for photosynthesis. Here, what I've represented in my slide is a leaf. So that transpire, leaves that photosynthesize. And so the photosynthesis is this essential process of exchanging carbon dioxide at the level of the pore that you've got on the leaves, which are called stomata. And naturally and unavoidably, that exchange at the stomata level of carbon dioxide go for a water loss. And so the situation that I've represented in my slide is a situation of a cool and wet air, where there's already a lot of vapor pressure in the air, and the gradients that drive water out of the leaves, because let's be clear, the stomatal chamber is saturated in vapor. So there's a gradient from the atmosphere driving water out of the leaves. In that situation, the gradient is small. Therefore, the photosynthesis comes at a fairly low water cost. But in future climate or in current climate, for example, here in Senegal in the dry season, the air is hot, it's dry, and there's a lot less water vapor in the air, which means that the gradients driving water out of the leaves are much higher. So photosynthesizing under those conditions is a lot more water costly. So that's basically a conundrum that the plant needs to resolve, is to maximize photosynthesis but minimizing water loss. And in all those years, what we have found out is a plant trait that's capable of restricting transpiration in that situation when the vapor pressure deficit, when the evaporative demand is too high. So today's talk, I'll be talking about the trait, what it does, what are the benefits of that trait in terms of crop productivity. And then I'll be talking about how we measure it, how we design a high-throughput phenotyping assay for it, and how we analyze the large data sets that come from the HTP platform. So let's get back a bit about 10 years back, when we are looking at transpiration. So that's here in the y-axis as a function of this evaporative demand, as a function of VP. And we compare two lines of per millet, one that was sensitive to water stress, and where the transpiration response to that increase in VPD, we were gradually increasing the VPD, that response was linear. So in other words, physically, mechanically, the demand became higher. The plant was responding by providing more water to the atmosphere. That second line, the black dots at the bottom, had a lower transpiration, and past a certain threshold of VPD, there was a breakpoint. So it was even giving less water to the atmosphere. So for us, I mean, what you've got to see is that the interest is in the difference between those two slopes, because they represent, between those two curves, because they represent water saving. The water savings comes from the fact that this plant had a lower transpiration rate and at a breakpoint beyond a certain point and use even less water. So that's basically those water saving that transpiration response to VPD represents. So after finding that in pearl millet, we tested, of course, a bunch of the species, a bunch of the germplasm in different species. And here is a rough summary of the type of transpiration response that we obtained. Again, so transpiration rate here in the y-axis, vapor pressure deficit in the x. Here is the situation of that sensitive line that I've shown before, so a very high slope, as opposed, for example, with a linear response, but with a small slope, which is also a type of restriction, and different situation in between with differences in intercept, no different in slope, but a breakpoint in between. Again, What is interesting and what matters here is the difference between those slopes represent water saving. Benefit number one, that trait confers an increase in water use efficiency. And so for the layman, water use efficiency is basically crop per drop. So more biomass produced per unit of water used. So what we did was a screening of germplasm of sorghum here in that slide. So with water use efficiency in the y-axis here. And then a number of germplasm tested. What you see is that we've got, a huge contrast, about twofold difference. And what we did was to test the extreme lines from that ranking, those which had low TE and those which had high TE. And we tested the transpiration response to VPD in 10 lines of having low TE, 10 lines of having high TE. And what you can see is those 10 lines having high TE have a lower slope. So they restrict transpiration. Those that are sensitive to VPD end up with a higher transpiration efficiency. And why transpiration efficiency is important? It's because it confers a yield advantage. And here I've plotted a few of the examples that we found over the years. Grain yield is in the y-axis in those three graphs, and transpiration efficiency is in the x-axis. And here I've plotted for you the case of maize, the case of peanuts, and the case of sorghum. So basically, transpiration restriction confer a high transpiration efficiency, which itself turns into the differences in yield. And very important one. The second benefit is that because the plants that restrict transpiration do not transpire as much as they could, and let's don't forget, we are not under stress here. We are under evaporative stress, but we are not under soil water stress. There's plenty of water in the soil. What those plants do, then they save water for later in the crop profile, in the crop cycle. What I've represented here is an example of cumulated water use as a function of time in two sets of chickpea genotypes that were either tolerant or sensitive, and each of those symbols represent about 10 lines in each of those groups. And so what you can see is that overall, those two groups of genotype use the same amount of water at the end of their cycle, right? So they are, but the slight difference that happens is the green group, the tolerant group, use a tiny bit less water here at the vegetative stage, and they use a tiny bit more water here at the reproductive and grain filling stage. So if I play around with those data and I do a bit of regression analysis between the water extracted here, which I put here as a, independent variable in the X, and I plot yield as a function of that in the Y, I've got a negative relationship. So the more water used here, the less yield. So those that restrict transpiration here would be those gaining yield. And those, the water extracted here, which is here in the x-axis, well, confers, a benefit in yield for those green lines. And here I've plotted what's different can be, for example, in a crop of sorghum when the grain filling doesn't take place well. Here is a line which had water during the grain filling. The grains are fully filled. Here, the plants didn't have enough water during the grain fill. The grains are poor or shriveled, and the flower sort of yield is very poor. Here is a modeling study that shows the benefit of that trait at a much, much larger scale. And it's in the case of soybean in the US. That's a study that has been done by Tom Sinclair with Pioneer at the time. What the left graph here shows the probability of success of that trait. It's red, probability of success is high, very high value of probability of success. On the left side is basically the yield benefit that I would have from inserting my traits in my soybean cultivars, distinguishing the wet years in the top here and the dry year at the bottom. And here is color coding for the yield benefits. We are in the red, which means that here in those dry years, I could expect something like beyond a ton in certain sites of grain yield benefit. Even in the wet years, it's still fairly positive, not everywhere. There's never a penalty, but of course the benefit is less. So with this, I hope I've convinced you that it's very important traits to put in breeding. And for the past 12 years, we have been basically looking for variants of that trait in for our breeding programs. So how do we measure it? So, well, let's get back to that initial slide. We measured transpiration as a function of VPD. So basically what we did initially was to grow plants. We put them in a growth chamber. We weigh pots to measure transpiration. So we weigh pots, we measure transpiration. And each of those dots here is a different VPD level. So every hour, we were raising VPD by a few decimal of kilopascals. So we raise VPD progressively. And after a number of those ladders, 8 in that case, we measure the leaf area because the transpiration is expressed here as a transpiration rate, which means the transpiration per unit of leaf area. So these are the 3 things that we need: transpiration, a high VPD, and leaf area. So here we are in a growth chamber. We can put at most maybe 50 pots in the growth chamber. So no way we can scale that out to cater for breeding programs. So we're thinking, well, how do we do that outdoors? So at the time I was in India, and India is a natural laboratory in the sense that there's a season where it's naturally high in VPD outdoors. So we had here a weighing station, so we could have transpiration. We were outdoors, we could have low VPD in the morning, we could have high VPD later on. So we organized the weighing very early in the morning, 7 o'clock, the second weighing at 11 o'clock before the VPD become too high, a third weighing at 3 p.m. in the afternoon, really covering the peak hours. Because if I get back to that graph, actually what we need is really a point here and a point here. And we would have most of the information of the response. And of course, we need to measure leaf area. What you can see here is that you've got plants that are bigger here and smaller here. And it's simply because here comes the logistics. We could not handle the 200 reels of a recombinant inbred line of that population in a day. So we wanted to have 4 replication. We had to stagger the planting in a way that we could handle the leaf area during one day, and then 4 consecutive days. And I shall say here that, I mean, good phenotyping is first of all good logistics. So we organize the logistics and the biological and biophysical requirement in a way that we can do that well. And it worked. Here, what we wanted to see is whether we would find a QTL for that trait. And we did, we found several QTL. And we wanted to see whether they would co-map with a yield QTL that was previously identified. And it did. It did co-map with that yield QTL that was identified, I mean, 10 years before. So that works. And I take that chance here to say that the reason why I've shown you those slides, so it comes back to a paper that Yana Polova published in 2012. We did that work, I mean, without the technology, but we had the most important things. We really really knew what we wanted to measure and why. And it's important to talk in that era of high throughput phenotyping, because the trend and tendency can be sometimes to put the technology ahead of the biological questions behind. And for us, we really put the biological questions ahead of the technology. We designed the technology and we took the technology that would address those biological questions. So how did we design then a high throughput phenotyping assay for that? Because of course, I mean, even if we could do it, I mean, it was like more or less unreplicable and difficult to reproduce to more number of crops. That could have been, I mean, simply a nightmare. So I get back to the layout. We need leaf area. So that's where, I mean, the initial contact was with Phenospex. I came aware of their scanners, the old version of the scanner. I think that was the F3 or 400, if I'm correct. And together with Phenospex, we basically designed a platform that was catering for our need, that was suitable also for the technology we would propose. So the scanners were here attached and positioned on an irrigation boom, and it was traveling on top of the plants. we don't see on that slide, but basically the platform is divided in a number of what we call sectors. In other words, experimental units. There was about 5,000 experimental units in that platform. And they develop, a 3D point cloud, out of which, that's what Gregor showed in his presentation, a number of plant parameters are extracted. And the one we were especially interested in here was the leaf area. The second thing was to measure transpiration, for which we needed to weigh pots. And so we set up a prototype of load cells. I wanted to check that we were able to do it really in the outdoors with that kind of technology. And so the prototype of load cells was weighing pots, automatically every hour. And here we use pots that were covered with beads to avoid soil evaporation. I'll get back to that later on. So here, with the scanner, we had leaf area. Here we had plant transpiration. We were outdoors, so we could be exposed to the high VPD condition during certain times of the year. So the type of data that we generated was, of course, very different from those responses we initially had in a gross chamber context. What we did, the first experiment that we did was to basically take those same genotypes and then test them on the prototype of the platform. And so here, what we generated were transpiration profile. So each of those peaks represent a day. And I've enlarged a few of those days here. And we basically reproduced what was expected from those two genotypes. So the one with black dot is the one that has a restriction here. The restriction comes in the form of flattening of the curves in the middle part of the day. Please note that the rest of the time of the day, it's essentially the same. The one with the white dots, terminal stress density line here with a restriction, unrestricted transpiration, has a much larger peak. And so when we talk about those water saving, they are represented by that tiny but substantial water saving and difference between those two curves here in those few hours of high VPD every day. So after that, we were happy that I was happy with what we could find. We tested that in also the crops, so it worked really well. So we developed, a larger part of the platform. You've seen that slide before. Here we've got 1,488 load cells. The time frame for weighing was 15 minutes. It's outdoors, and so it's waterproof. And basically, we're ready to cater for the needs of breeding programs. What we did also was to shift from small pots and to shift to larger trays where the crop would have more like a canopy structure like in the field. So I would almost consider those trays as microplots. And here I've taken the example, this is the chickpea crops where we have 2 rows and 4 plants in each row. And we're using the density of planting of chickpea in a natural field, about 30 plants per square meter in that case. So now with this, we change scale basically from 50 pots in the gross chamber, 50 in the prototype to 1,500 load cells. And so the big question then was, how do we analyze those large data sets from the high-throughput platform? And so it's important here to frame the question. So here was what we were measuring here. We were measuring slopes, breakpoints, intercept. Here we are in natural situation. So what features did we need to extract to characterize that transpiration response? And so here, of course, we think of the peak and we think of a number of things. I'll get back to that later on. The thing is, how do we get transpiration profiles that are clean? What you see here, it's, we've got sort of a hole sometimes, it's a bit rocky in some places. So we need to develop something that would generate smooth transpiration profile. And here were our first sort of steps into developing this. Here I've plotted the VPD of a few days that we were sort of flowing up. And here was a few of the transpiration, evapotranspiration profile, and because we didn't have the beads on top of the tray, so that we measured plant transpiration plus soil evaporation, so it's evapotranspiration. What you can see is that, well, it's nice. I mean, so the system is sensitive, but we've got noise. I mean, we look at, for example, the gray dots. It's basically one single tray. And you see that in the middle part of the day, it's not that smooth profile that we wish we would find. Here we've plotted the Penman-Monteith equation. So Penman-Monteith is a representation of the potential evapotranspiration for a crop during a day, given the climatic conditions at the site. So we use that as a nice benchmark to be sure that the data we were generated were in line with what the biophysical conditions would enforce. So here, what I'm going to present you is a work really typically of the paper that Grégoire announced. That's a work of Subhashri Kaur, who is a bright young computer scientist whom I met at TICRISAT when she was doing her PhD, and I became her supervisor for that PhD. She's now a postdoc in the US, extremely skilled, and she was basically like the computer brain behind that EasyTR pipeline, as we called it. And I was basically the one trying to make a biological sense in that pipeline. So I would say it has been, a very nice journey with Soumya to work at the confluence between, a plant biologist, myself, and a computer scientist on the other side who didn't understand much of biology, and I didn't much understand about computer coding and R scripts. We've got three legs in that pipeline, and I'll present two of them today. The first one on the left, which consists in extracting transpiration profile. The second, which is the stage where we extract the features, and the third one, I'm not going to talk to it about it today for the sake of time, which is about breakpoint and how we go into sensitivity analysis of those profile responses. Essentially, the steps of extracting transpiration profile goes by first the cleaning of the data, the load cell data first, even before we compute the raw evapotranspiration profile. There's a second stage of cleaning, which is empirical and is based on what I was saying, the Penman-Monteith equation. I'll show you what it consists in. And then the third stage is a stage of smoothing those profile. The fourth stage is a step of extracting transpiration from the evapotranspiration profile. So let's start by that leg. To do that work, we use the data set where we had well-characterized information of a data sets of a set of germplasm of chickpea, well-characterized. So we knew the kind of germplasm we had. We had high TE line, low TE line, we had the wild germplasm. So in other words, we used a set of data where we could sort of expect response, and it was very helpful to sort of anticipate what kind of response we would get, and then whether because of course we tested a lot of things during the development of that pipeline, and whether we considered that the pipeline was sensitive enough to extract the information from those profiles. So here is first step. Here are the weights that are generated by the load cells as a function of time. And here is basically what you have, as a regular sort of daily decrease in the weight because of plant transpiration. And here is a night, and here is a day again. And those big sort of jumps is when we irrigate. And so when we irrigate, we've got a big jump in the weight and a big jump down of the weight when the tray drain because they lose water afterwards. So the first leg of the cleaning was about extracting those big spikes. And so in that profile, that first sort of cleaning stage took out all the parts that are in black. So all the data that are here were taken out, only remains the red one. Second stage was to generate the ETR profile. So they are represented here as a function of time. We've plotted already Penman-Monteith here in the dotted lines. And here I've represented 3 cases of profile. So you see that it's fairly rocky. It's fairly rocky, and the reason why we use Penman is that we had that kind of things here. we've got an evapotranspiration that go beyond Penman. Well, that's not possible. Here, for example, that's not possible. So we use Penman as a kind sort of safe benchmark to clean out whatever was going out of the Penman-Monteith curve. So we wanted to have data that would remain under the curve. And for example, all those big negative data that we had sometime, of course, they were outliers and they were cleaned out. Then the next step from that was a smoothing step. So we have still here Penman-Monteith, and we generated those smooth profile. Here, I don't enter into the detail, but Sulia tested, a bunch of possible smoothing techniques until she found this one. And there's more detail in the paper. So there we came at a stage when we can start comparing the ETR profile in those groups of chickpea cultivar that were well identified. So in the black dots here is the VPD. And in the yellow, the green, and the blue, we've got the three groups of genotypes. And as expected, you have the high TE here, which are like these big peaks that, like what we saw before. The wild or there, we didn't know, but we pretty much expected them to have a transpiration that would be restricted in the middle part of the day. What I want to show you there is that with that smoothing, we had a good sort of distinction between the genotypes. And the last step of that pipeline was to extract the transpiration rate from the evapotranspiration rate. So in other words, we wanted to kick out the part of the soil evaporation, because don't forget, in the chamber, we could put beads on the top of the pots and basically zero soil evaporation. We could not do that with 1,500 trays. So we had to find out, what was that soil evaporation component in another way. We use that equation from Novak and Avrila, which gives basically transpiration as a function of the leaf area index. In the platform, we measure leaf area index. So we had a measurement, regular measurement, the daily measurement of the leaf area index, and we use that to extract and to expel the soil evaporation. And currently with Sumia, we are working on different data sets to adjust and correct possibly that beta coefficient, because here we used it as generic for different crops. We think that there's possibly slight variation among crops. And so we generated data where we could sort of adjust that beta coefficient for different crops, but we're working on that paper at the moment. Okay, so second stage of that pipeline, we have now smoothed transpiration rate profile, and we get back to that situation of what kind of features do we need to extract compared to what we had before. And so before we basically sit on the chair and thought, okay, well, we are gonna have like the maximum transpiration here, that's gonna be important, and we are gonna have like maybe the slopes between the highest peak of the hour to get to that stage, and maybe the area on the curve here, and maybe the whole area under the curve here. Anyway, we listed a list of about 15 features that we think were representing well the transpiration response to VPD and their differences between genotypes. And so here is that list. You'll have the recording of the talks, so you can have access to it, and anyway, it's published. After that, we tested whether we could cluster the genotypes again according to that set of features, and we could again distinguish those three clusters that were essentially representing the low TE group, the high TE group here, and the wild cultivars here. So we're confident that the set of features were actually representing well the expected distinction between the cultivar in that set of lines. So after that, Sumia designed a way to identify and distinguish features according to importance. I would not be technically able to explain you the difference, but basically that's called the mean decrease in Gini. And basically when that mean decrease in Gini increases, it means that the features have a high importance. So in other words, explain a large part of the phenotypic variation across the different features. So that was good because out of the 15 features, I mean, essentially we had 9 that were, I mean, not very, very important. And so that left us with 6 features that had the most important. And among those were, I mean, I would say, those expected one. I mean, the max transpiration, the area under the curve in that peak, those peak few hours of IVPD, the slopes between the minima and the max. And so what Surya did then in the pipeline was to develop an extraction features where we extracted here the means of each of those 6 important features, and then, well, for basically statistical analysis of the differences. And here, each of the color groupings represent different significant groups for those different features. So that's all I wanted to present today, but in quick conclusion. So I hope I've convinced you that that trait is an important one to measure and to breed for. I've shown you that we can measure it at a high scale, and I've shown you, I mean, what we have put in place to do it. And we can now analyze those high-throughput data sets, and we can then deploy it in breeding. Thank you very much, and I'll be happy to entertain your questions.
Thank you very much, Vincent, for this excellent talk. And certainly this was a depiction of tremendous labor that has been done, despite having the right technology at hand. So for now, then, I want to open the discussion. So feel free to ask questions to Vincent and Gregoire. And please use our chat function of our Zoom webinar in order to let me know which questions you have, and I will speak them out loud and ask Vincent. So, Vincent, one of the questions comes from myself. Maybe I will give the audience some time and start over with this question. So, as you have outlined, your data supports that plants which use less water during the early stages of development have advantages, especially during the grain filling stage, and hence also have higher yields. How does this relationship hold through different degrees of resource limitations? Yeah, either water or nutrients even. Have you looked into interactions of your selected crops or high ETR crops with other environmental factors or even stresses?
Yeah, thanks, Philippe. It's a good question. I mean, it's basically the key question about G by E by M, and genotype by environment by management interaction. I mean, let's be clear on one thing. I mean, when we worked on that trait. I mean, I was working for ICRISAT and working for the semi-arid tropics. So we were basically like targeting environments that are extremely water limited, and where water saving in those environments turned out to be very, very important factors. So for example, in the case of chickpeas, where we had a crop that was grown under residual moisture with virtually no rain during the season. And so was the same for sorghum. So we had situation, and that's why I've shown an example of modeling studies. We had an example where that trait had a value. But of course, we have different situations where that trait that limit transpiration, so therefore limits photosynthesis, would have a negative effect. In those cases where water is limiting but not that limiting, then we would prefer to breed against that trait. It's interesting, for example, to find that in the case of rice, I mean, was in close contact with my colleague Amelia Henry from Erie. And then she was saying, well, it's interesting because in our case, I mean, we find that those that don't restrict transpiration or those that are our elite, and for good reason. I mean, she was working on rice in an environment where VPD was probably much less. And we know that rice has a poor root system, and basically rice that transpires is good rice. And so there, I mean, she probably needed the contrary than what we did. So yes, we are aware of that limitation. Actually, it's not a limitation. The platform that we've developed would cater for the same if we had to breed out. So we could select against, or we could select for that trait depending on the need. But yes, it's an important preliminary question, which of course we had.
Yes, trade-offs are very, very likely to happen if you observe the maximization of one certain physiological variable, then often a different one has to pay the price for that. And this in turn, of course, also reflects back to how suitable a certain crop breed is for a certain very peculiar environment. And I think also there in India, you have a multitude of different environments and therefore a high demand of different sorts of adapted crops. Thank you very much, Vincent. The next question comes from Filippo. He asks, or he says, first, thanks for the great presentation, Vincent. I'd like to ask if you measure the yield of the different genotypes of the experiment and if you find any relation between yield and the transpiration rate.
Alors, so we have several data sets where that connection was made. If you look at the paper Zamanala et al. 2011 in chickpeas, we did measure that trait indirectly in the lysimeters in big tubes, where we also measured grain yield. So we had data on those showing a transpiration restriction or a kind of, I mean, something akin to it, and grain yield increase. So we had that relationship. In other cases, things were indirect. We are, currently finalizing a paper in sorghum where we have established that relationship between transpiration rate, transpiration efficiency, grain yield increase. And so direct measurements are not easy because the platform was catering for a transpiration response to VPD, but was not there to apply any kind of stress. So it's difficult, let's say, to measure yield in the same situation where you measure the traits. It's a matter of basically joining the scales.
Thank you very much.
But basically what helps us make the connection is crop modeling.
Certainly, yes. Thank you. Ben Gruber asks, what types of factors or what types of influences were causing the large deviations in transpiration outside of what was expected from Penman-Monteith, i.e. the effects that had to be removed in the analysis pipeline?
Alors, so the reason for the fluctuations is, first of all, we were outdoors with all kinds of possible disturbance, micro variation in the conditions, wind, birds, people walking on all sorts of things were possible there. Because the load cells are very sensitive, and so anything could alter the weights. But it's also, an electrical signal that's, very, very fine, and that can also fluctuate at some point for unknown reasons. So we had a part of known and a part of unknown reasons. By and large, it works really well. Now that I've developed, a smaller platform at IRD, and we work then in a growth chamber, not in a growth chamber, in a glass house. What we can see from the platform indoor is that the data are a lot smoother, a lot cleaner than what they used to be outside. And so I guess that's being outside that created all those fluctuations.
Yes, that's the joy of going out and doing field research. You have a lot of noise that you cannot foresee, and of course that even the most detailed models do not account for. The next question from Alex is, has their high transpiration efficiency trait already been realized in the modern germplasm, in chickpea or sorghum, or unconsciously bred for? So is there still potential for this trait in commercial breeding, I guess?
Well, there's clearly a potential. And I compared, for example, the top germplasm we had in sorghum for TE to the elite cultivar developed then by the sorghum breeding programs. And there was clearly an advantage. For example, West Africa sorghum breeding based a lot on Guinea land races. If you look at the 2011 paper where we measured that germplasm, well, the TE of those Guinea land race of sorghum was much lower than the TE of, let's say, Kafir or Dura land race of sorghum. So in other words, there's still a huge benefit to get from breeding that trait into different breeding programs. We did crosses, in fact, and so there's a number of backcross nested association mapping populations of sorghum that have been developed using some of the donors that I've presented today in my TE screening and elite materials. And well, basically the work is ongoing. But you are right to say that there could have been also, I mean, unconscious selection for TE, provided the selection pressure was properly done, and provided the breeding trials were done in seasons when BPD was high and was pushing for lines that were bred actually for that trait.
Alex is following up on this. Any idea on the underlying genes of the TE trait?
That's part of, let's say, of the more fundamental work that we have undertaken. I've not talked about that today. But the reason for the transpiration restriction is basically where we want to go. And there, I mean, the first, well, I would not like to call candidate because it sounds a bit like a magic bullet, and I don't like that idea of magic bullet. But basically, let's say the domain where we wanted to invest and we are investing currently is to understand, I mean, how water channels through plant tissues and especially through root tissues to reach the xylem and eventually reach the leaves. And so one of the facilitating factor there is aquaporins, or it's membrane transporter that facilitate water movements through membrane, and that has a role on the conductivity of plants to water. So that's one of the things, and we've got evidence, in fact, that we've got differences in aquaporin gene expressions in lines that were either sensitive or non-sensitive to the high VP. So yes.
Thank you, Vincent. Going back from the very complex to a little bit more basic of a question, Gustavo asks, Vincent, can you please explain the advantage of adding the leaf area to evaluating plant transpiration?
I mean, the short answer is that that's basically what would allow you to compare Arabidopsis and maize. It's basically the problem that you face when you compare plants that have, I mean, very, very large difference in canopies. And so it's important to have like a common metrics to compare genotypes that would potentially have very large differences in leaf area. So in other words, we want to get rid of course, strong influence that leaf area would have on the magnitude of the transpiration if we were only comparing transpiration. And well, the thing I wanted to say, but I forgot.
Well, it's basically a means of standardizing the transpiration
yes.
rate.
Oh, yes, I remember. I mean, the second reason, very important one also, is when I got into that business of trying to understand transpiration of plants in under high VPD, so in the middle part of the day. So initially, I mean, what we were using or trying or wanting to use were porometers. And so with porometer, you clip the portion of the leaf, and then you get a measure of the leaf conductance. But I quickly realized that that was a nightmare. I mean, it's extremely variable, you know, exposure to light, leaf orientation, all that sort of things, and no way to scale that out. So I thought, well, leaf conductance is basically like a transpiration per unit of that small clip of plants of our leaves that you take. We could do that at the leaf level, at the canopy level. And so the transpiration rate, what I call canopy conductance. I know that there are certain people that don't like it because it doesn't reflect the heterogeneity through the canopy, but it gives a good average, I would say, and it's robust. I mean, it's extremely robust, and you can measure that, and plant-to-plant variation is very limited.
Yes. Alex G is asking, have you looked into structural differences at the leaf level in, for example, stomatal density, number of leaves there, whatsoever?
So if you look at Kolova et al. 2010, so that's Jed's bot, we did that. We compared those two lines that I've shown today, I mean, the sensitive and the tolerant line, because we thought, well, we could have differences in stomata density, but we didn't find anything. So we have basically like given up on that route later on.
Mm-hmm. Great. Coming back to the earlier question from Gustavo, actually, Vincent, you by now have an extensive experience throughout your whole career on plant water use efficiency and also the use of phenotyping equipment. So in respect to the methods you use for your research and the quality of the methods in the past 10 years, how would you describe the technological development and its influence on quality of your research, especially?
I mean, clearly, I mean, we could not phenotype for that trait at a very high rate without that technology. I mean, that's the example I have given. You know, we could do it, but we could not scale up. So in other words, I mean, measuring that transpiration rate without something that gives you leaf area, even if not totally exact, but quick and consistent, is essential. And then weighing pots on more than a few hundreds is simply impossible. So having that technology, and I would say the original version of the scanner was enough for that. But having the technology there was essential, yeah.
All right, thank you very much. So I have a question for Grégoire as well. Grégoire, coming back to the beginning, you introduced the new PlantEye, F600, and you expressed that in this iteration of your phenotyping sensor, that both speed of scanning and speed of the integrated analytics that extract the features from the 3D point clouds has improved. So can you tell us a little bit more on what other features may have improved or may have been added even in this iteration of the device? Can you do more with it than before, or just everything a bit better?
Yeah, so the last 10 years at Phenospex, we were really focusing on the hardware and really developing a sensor that is really designed to assess crops. And I mean, we talked already about outside. I mean, measuring a plant is, first of all, quite complicated, especially if you go outside or in greenhouses. You have a lot of environmental constraint, you have light, a lot of things. And for us, it was clear, if we want to have a sensor for plant scientists, we need to, to have it, that it can be used under any circumstances. For us, it was absolutely not acceptable that you have to drive plants to an imaging chamber to shape the plant. And if you have a sensor out there, it should work under all these conditions. And that's something we really worked on hard. And I would say this, what I said at the beginning, this 10 years resulted at 600. And that's really a sensor on which we can really now build up. Because the data is now really in such a quality, what we were always looking for concerning the resolution, but also concerning the sensitivity and the resistance to sunlight, that now we are actually, and that's why this presentation is also so cool, that we now move to the next level, which is a data analysis. So at this moment, we can extract from the PlantEye and from these gravimetric platforms about 20 parameters or features. But now we want to build up more. Especially because most of our clients, obviously, some of them are interested in maybe leaf area or are interested in biomass, but often people are interested in more integrated parameters like healthiness of a plant, like scores, etc. So what we now do is that we invest in actually extracting more features from these 3D point clouds. Also offer to our clients tools and software which are using machine learning, artificial intelligence, in order to actually develop models to fit their traits they're looking for. And that's where we're going now. And that's why also it's so interesting to see also our, like Vincent, who is using our equipment. He's getting this data, but actually he's working on this data and creating new features, which now we can integrate also into our platform, made it available to the entire community. And that's now where we're going to be heading to in the future as a company. And by the way,
I can see.
By the way, one thing, maybe a small announcement, which is also related to the question of Alex G. Structural parameters, how do they affect actually things like VPD? That's something we're going to be looking in now. So together with Vincent, we are having a PhD, which is exactly investigating these interactions and also developing new features or new algorithm to extract additional features, which are also giving some insights about structural parameters of the canopy.
So there will be new functionalities, new features coming up soon, as R&D is being targeted towards this within Phenospex. Good to hear, and also good to hear that your strategy of automated feature extraction is possibly also making plant phenotyping accessible to non-academic researchers on a broader scale, and maybe also getting the transition to other business areas where plants are of center and of interest out there.
Philip, maybe I would like to add something for the semantics, because there's so many expertises involved in this plant phenotyping. It's good to have the semantics, right? So what I mean with features is really like data, what we extract from these point clouds or what we extract from these scales. Mm-hmm. So these are really independently computed features. And these features are then translated by the biologists into traits. And that's exactly what we're going to be doing in futures, that we build software to enable the client to translate the features what we provide, independently computed parameters, and to allow our clients to actually build their models and train their models in order that they can predict their traits. So this is the difference between the features and traits. Just wanted to have this clear.
Okay, so do you see in the near future business or use cases that you can yield now with the F600 that were not possible with the older generations of PlantEye?
Yeah, I mean, first of all, I mean, the big advantages are higher sensitivity I mean, now we're going really into technology. So what we did, we improved the resolution of the scanner. So basically, we actually developed our own computer chip using FPGA. So we actually can read much quicker the cameras. So we are faster, and we are 7 times faster and have 40% more resolution. And also we improved the camera chips. So we are more sensitive. So for instance, if you're interested in traits which are correlated to stress, to healthiness, we are just more sensitive, and we can actually capture and quantify much earlier when a plant is stressed than before. So this is gives you, this is kind of advantages with the new PlantEye F600. And additionally, also what is interesting, we actually added a lot of APIs, which makes it actually easy for our clients to integrate these sensors into the existing infrastructure. So there's an API, the PlantEye has also a face. So the PlantEye itself has now even an interface, which shows you actually what the sensor is doing and makes all this integration process much easier.
So a lot of accessibility features for non-academic scientists or corporate scientists, for technologists and engineers to integrate this feature. Coming back to the hardcore researcher, Vincent, would you imagine benefits of the new model for your, particularly for your research? Gregoire was talking about increased resolution. So will this automatically maybe also increase the outputs of your, or the turnouts of your data and its accuracy?
Well, I would say, I mean, we can't jump from one trait to another, like you change shirts. So it's I mean, we are going to continue on the leaf transpiration response, and of course, I mean, the trait and the feature, well, the feature that we need to get from the scanner still remains the leaf area, and the features we need to get from the load cell remain still transpiration related and maximum transpiration, etc. So I mean, the focus of the work so far is still going to be, I mean, focused on this. I mean, of course, yeah, the higher resolution can only make it better. Over the last few years, we have also discussed, I mean, what Greg was talking about on plant architecture. We are getting a lot more interested in plant architecture, 3D architecture of the canopy, and there are certain features of the PlantEye that characterize that architecture, and we like to go beyond that. So we would like to extract new features that characterize the plant's 3D architecture. And so, of course, that resolution, a better resolution that the new scanner has, is there to serve us. I mean, of course, I mean, there are certain things that are potentially usable, given that we've got multispectral sort of signaling, is everything that deal with nutrition, nitrogen, looking at colors, and everything dealing with disease effect. It's not my topic, but I can see the potential for those that would be interested. So it of course, I mean, opens a lot of opportunities that are beyond my own field of interest, but that are there.
So I want to thank you both, Vincent and Gregoire, for today's episode of the Phenomics webinars and the very well kickoff of our season 2. There will be coming more in the near future, and our next Phenomics webinars will be in about 1 month, be given in the same format, on water use efficiency as well, and finding the best trade-off between productivity and survivability. It will be given on October 28th in a live format again at 1400 CEST. And our speakers will be Karen Marchelion from PlantEye and Alexis Comar from HiFen. Thanks again, Vincent. Thanks again, Gregoire. I hope you enjoyed it so far, and thank you also into our audience for the suite of questions. Have a good day.
Yeah, thanks a lot. Ciao, Philip. Ciao, everybody. See you soon.
Thanks, everyone.